We are going to study about blotting techniques. So first of all, what is blotting? So blotting is a technique of transferring DNA or RNA or proteins onto a carrier, okay, onto a carrier so that they can be separated, and we often use gel electrophoresis to see the bands, okay. So just discussing about the basic. What are the types of blotting techniques? They are southern blot, northern blot, and western blot. Southern blot is used to detect the DNA, northern blot is used to detect RNA, and western blot is used to detect proteins. So, what is the history of southern blotting? Let's start one by one. So, southern blotting, it was named after Sir Edwin, and this technique was developed in 1975. So, certain blotting, it is used to detect the DNA, and this method involves separation, transfer, and hybridization of the DNA. So this DNA can be a single gene, or it can be a part of a larger piece of a DNA. It can be a fragment of the DNA. Okay, and the main method of southern blotting is hybridization. So, what is hybridization? Hybridization is a process of forming a double stranded molecule between a single stranded probe. We will design a probe and we will allow it to bind or to form double stranded with our target DNA. To find out the target DNA, we will use the single stranded DNA probe, which will be designed by ourselves, okay, and a single stranded target DNA. Whether the target DNA is present in the patient, we will design a probe complementary to it. So what is the principle of this southern hybridization? So the mixture of the molecules, like the hybridization, the double stranded DNA which is formed after hybridization, the mixture which is formed is separated on the gel electrophoresis, separated through the method of gel electrophoresis. How they are immobilized on the matrix? So first of all, what happens, that the mixture of the molecules are separated, okay. Say for example, it is fragmented, a long segment is fragmented, it is immobilized on a matrix, and then the probe is added. The probe is designed by ourselves to the matrix to bind to the molecule. The probe, if we will design the probe, and just to check whether the target DNA which is which binds to the specific probe which we have designed, whether it binds to the specific probe or not, okay. Whether the target DNA, if it present, then there will be binding to the probe, otherwise not, okay. So probe addition to the matrix to bind the molecules, and the unbound probes are removed. Only those probe will bind which have the target DNA present in the patient, and other will not bind. Okay, so the place where the probe is connected, it corresponds to the location of the immobilized target molecule. Okay, so wherever the probe is located, there only you will find that the hybridized DNA, that hybridization has occurred. Simply, you can understand in that way. So, what are the steps followed in southern blotting? At first, the DNA is digested. We will get many fragments. The fragments are then separated on gel electrophoresis. They are then transferred onto a membrane, okay, basically nitrocellulose membrane. Then the probes are added. If the target DNA is present, then there will be double strand formation, otherwise not. Then they are visualized on autoradiogram. So this is how the process goes on. It is like the DNA is applied to the gel. Okay, how we will start with? So first of all, DNA is applied to the gel. Through gel electrophoresis, there will be migration of the DNA. Then what will happen? It will be transferred onto the nitrocellulose membrane. Okay, salt solution are used. They will blot the DNA onto filter by capillary action. Okay, so blotting will be done by capillary action onto the nitrocellulose membrane. Then the DNA is transferred to the filter. Okay, so after which, after transfer to the filter, then hybridize, see this filter paper have all the DNA. Okay, then the probe are added. So if the target DNA is present, the probe will bind to it and there will be hybridization. So probe hybridizes with the complementary DNA sequence and then they are visualized through autoradiogram. Understood. So, what are the applications of Southern blotting? Why we use Southern blotting? Southern blotting, it is used in gene discovery, in mapping, in evolution, in developmental studies, in diagnostics, in forensics. Basically, to check whether the target DNA is present or not, to find out the parent of a child, and so on. So, next, starting with Northern blotting. So northern blotting, it was developed by James Alvin and George Stark at Stanford University, and northern blotting, it is a technique for detection of specific RNA sequences. So what are the steps involved in northern blotting? At first RNA is isolated, then RNA is loaded onto the agarose gel, again same blotting on nitrocellulose membrane and labeling it with probe. So washing to remove any unbound probe and detection by autoradiogram. So simple, moreover all these steps are same here, just here it is done for RNA. So RNA extraction from the sample, electrophoresis, then they are RNA are separated by size, then they are transferred to the nitrocellulose membrane. Probes are added, the probes are here labeled and visualization of labeled RNA on X-ray film. So this is about northern blotting. So what are the applications of northern blotting? So, a standard for direct study of gene expression at the level of mRNA. So, gene expression study at the level of mRNA is for that northern blotting is used. So, detection of mRNA transcript size, the size we can understand, we can know about the gene expression, if that gene is expressed or not, what is the size of that mRNA. Study RNA degradation, whether that RNA is present or not, whether it has degraded. Study RNA splicing, can detect alternatively spliced transcript. Okay, so normally spliced and alternatively spliced transcripts can be detected. RNA half-life can be studied. IRIS, which is internal ribosome entry site, is also studied to remove possibility of RNA digestion. Okay, versus second cistron translation. So these are all the application of northern blotting. Now coming to western blotting. So we studied the history for northern blotting and southern blotting. What about the western blotting? So Dr. Douglas, the Lake of University of Arizona School of Medicine's Department of Microbiology and Immunology, he have discovered western blotting. Okay, so what is western blotting? So a Western blot, it is a technique in which proteins are separated by gel electrophoresis. Okay, so proteins here are they are separated by gel electrophoresis and transferred onto a membrane sheet. So first was for DNA, this is for RNA, now this is for proteins and they are transferred onto the membrane sheet. Okay, and the specific protein is then identified through its reaction with a labeled antibody. So there first of all DNA was used as a probe, and in the third case here through its reaction with a labeled antibody is used for hybridization, not hybridization, for the reaction. And so this principle works on the so this technique works on antigen antibody relationship. So what are the prerequisite for western blotting? So the SDS page technique is mainly used for western blotting. What is SDS? It is uh, a kind of detergent. The full form of it is sodium dodecyl sulfate, which can dissolve hydrophobic molecules. Okay, and these are also negative charged because it has a negative charge sulfate attached to it. So what are the steps in Western blotting? At first, gel electrophoresis is done where the proteins are separated according to the size. Here SDS is used. Then transferred to the membrane by applying current. Same here, nitrocellulose membrane, the proteins are then transferred, the bands are then transferred to the nitrocellulose membrane. Then blocking is done. Blocking is done to prevent non-specific protein interaction, okay, between the membrane and antibody protein. So antibody is added to it and then it is blocked so that non-specific interaction will not be done. So you can see here protein blot on nitrocellulose membrane. First of all, STS gel where proteins are separated based on the size. Then they are transferred onto nitrocellulose membrane. Then antibodies, they are labeled with specific antibody, and then blocking is done so that non-specific binding is not there. And then antibody have a fluorescent tag attached with it. The fluorescence will be then revealed. Okay, so what is the application of Western blotting? So Western blotting is used to identify specific proteins or their masses, and the confirmatory HIV test is also detected by the method of Western blotting. Uh, their anti-HIV antibodies is used in a human serum sample. If the person have HIV, then their blood sample will also contain anti-HIV antibody. So if anti-HIV antibody is present, then the person have HIV. Okay, so this test also uses the principle of Western blotting, and the definitive test for bovine spongiform encephalopathy, that is BSE, commonly known as mad cow disease, can also be studied or can also be identified through Western blotting, and some forms of Lyme disease testing, they also employ Western blotting. So this is a very basic about the three blotting techniques. If you want more detail of it, you can write in the comment box, and please don't forget to like and subscribe my video. Subscribe to my channel and like my video. Thank you.